All right. Well, welcome back, everyone. If you are here, we are about to start the native seed production concurrent session, and we're excited to have the following speakers present this term or this time. And I would invite Curtis to go ahead and start sharing your screen. Um, our first speaker is going to be Curtis Drake, and he is from the Colorado Natural Heritage Program. And he's about to talk about creating a phenology model to inform native seed collection efforts. So, all right, take it away, Curtis.
All right, thanks, Morgan. So, let's see here. All right, so I'm going to start out by giving a little bit of background into our seed collection program so you can understand where we're coming from. So the Region 2 Native Plant Materials Program, it began in 2022, and it's a cooperative agreement between the Colorado Native Plant Society, the U.S. Forest Service, and the Colorado Natural Heritage Program, which I work for. And so Region 2, that's the Forest Service Region 2, and you can see all the areas it covers on the map on the right, covers Colorado, Wyoming, South Dakota, Nebraska, and Kansas. And the purpose of this project was to beef up the seed supply that the Forest Service has for its forest units to use in restoration. Anything from post-fire restoration to restoring disturbed areas such as ski resorts or campgrounds, or revegetating roads that have been decommissioned. And I think part of it also has come out of the need for more seed in response to the larger wildfires that we've been having in Region 2. And you can see it's a big area to cover. And you see that little flag there, that's about where the Colorado Native Plant Society, or sorry, the Colorado Natural Heritage Program is located. So um, it's a big area to cover. We can't exactly drive all the way up to South Dakota to take a look at the plants and figure out if they're ready to collect or not. So that's why we've been thinking it'd be great to have a model to help us with that. Um, so, and sorry, I just got lost my train of thought. Um, within Region 2 as well, we use we have a lot of different ecosystems and provisional seed zones. So if you look on the map on the left, that's a map of the different provisional seed zones as they were published by Bauer et al. In 2014. And you can see there's a lot of diversity in there of both ecoregions and climate with the different ecosystems we have and the different elevations. So that's kind of our goal, is to fill out those um, the seed supply so that we have seed for all of the different places that we need. And another thing that makes knowing when to collect seed in our region difficult is for anybody who's in the temperate region, they know the weather changes a lot from year to year. It's not exactly predictable. So your phrenology is going to change from year to year as well over a period um, of from days to weeks or maybe even months, where it'll be different from one year to the next. Um, Once we've collected seed, we send it to a USDA nursery in Coeur d'Alene, Idaho, where it's grown out and increased, and then that seed is regionally owned by Region 2, and it can be bought by the Region 2 units to use in their restoration projects, however they'd like. All right, so for the model, we have a couple different types of data that we're using to create this model. Um, the first type is the data that we collect. It's these point locations um, of places that we've visited, like the photo on the left, where we know there's a great potential seed source there, a large, healthy natural population on Forest Service system lands. But when we're there, we may not collect. We may or may not. If we're there a little bit too early and the seed is still immature, or if we're there a little bit too late and it's already shattered, those are reasons why we wouldn't collect. Um, And while we're there, what we always do is we collect categorical phenology data, which you can see here. We have 5 categories, percent, and they're just estimates of the population. So the percent of the population that's vegetative or developing flowers still, the percent that's flowering, the percent that's developing seed, the percent with mature seed, and then the percent that's already post seed dispersal. And the other type of data that we collect, well, that we use in the model, but we don't collect ourselves, is the climate data. So we don't collect climate data, but there's a lot of people out there who do, luckily. And the Prism Climate Group um, collects data from all these different networks of weather stations. They have over 25,000 different weather stations in their network, and those include all of the major NOAA networks, the U.S. Forest Service, Bureau of Reclamation, USDA, and many others. So they have all these point locations and they feed them all into a sophisticated computer model to be able to create like a continuous layer that goes over the United States and Canada, like this one that you see here on the left of average daily temperature from February 1st to 15th of this year. And their model takes into account a lot of different things such as elevational effects, temperature inversions, and terrain features that cause rain shadows. And this, like I said, this data is publicly available and it's available at a 4 kilometer and 800-meter resolution. So that resolution, we use the 800-meter resolution and it happens to be a pretty good resolution for like estimating what a plant population might experience. So that's the climate data that we're using. And we use that plus our own point data, phenology data to create a growing degree day model. And that model is defined in the equation you see at the top. I tried really hard to make this slide look more interesting, but I just couldn't make this math look interesting, so sorry about that. But you can see the first 2 terms in the equation are maximum temperature plus minimum temperature divided by 2. So that gives you an average temperature for that day. And then you subtract that from the base temperature, which the base temperature is the temperature at which or below which you believe a species will not grow at all. And so for our model, we used 2 different base temperatures. We used 50 degrees Fahrenheit for warm season species and 32 degrees Fahrenheit for cool season species. And then there's a couple of things about the model. The maximum temperature variable here cannot exceed 86 degrees Fahrenheit because the theory is that plants will, above a certain threshold, they'll have a linear increase in their growth rate up until 86 degrees Fahrenheit, and at which point it'll plateau. So if a day has a maximum temperature above 86 degrees Fahrenheit, then that term is just recorded as 86 degrees Fahrenheit. And then also you can't have, if this equation solves to be negative, it's just recorded as a 0 because even if it was colder than that plant could have grown on that day, it doesn't go backwards in its phenology. It can only go forwards, or it cannot move, but it won't go backwards. So if that equation solves to a negative number, then it's recorded as a 0. And then so that's the growing degree day accumulation. So you take that equation and you add up, the result of it over time. And then we also calculated a target growing degree day accumulation for each of our species. So we took our phenology data and we found the earliest date that they were ready for harvest, which we defined as the percent mature seed and post seed dispersal is equal to or greater than 25%. So we found the earliest day for that and then found how much growing degree day accumulation had they had up until that point. And then we averaged that across our 3 years of data. And then that gave us like a target growing degree day accumulation that we're shooting for each species. And so the desired result is to have this nice application or interactive map where you have the different points where you're modeling to, and you have, if you can click on a point and you can see the species that is there, you can see what its current growing degree day accumulation is at, and then what its target number is at and what that difference is, to help you plan when to go out to that site. Now, I say that's the desired result because it hasn't really worked out yet. So when I wrote this abstract, we were on generation 1 of the model. And in generation 1 of the model, we wanted to generalize the results to the ranger district level, which you can see the different ranger districts here on this left image in the different colors of green. But that didn't work very well. Especially in the ranger districts that had big elevational changes, ranges, such as that point on the right. You can see it's in a kind of a low point in the terrain, and that's going to have a lot different climate than, say, up on that hill next to it. So that's why this year we decided just to model to a specific point, or that 800-meter grid cell that point happens to be found in, instead of trying to generalize to a larger area. And if this version of the model does work well, I suppose you could generalize to similar elevations within a similar latitude of that point. But trying to generalize over different elevations doesn't seem to work well. So I'm assuming a lot of you attended this talk because you're facing a similar issue about trying to decide when to collect seeds. So to keep this talk from being too disappointing, I thought I would share, what we're doing in the meantime without a model. And I will say that these lower tech solutions, they do work quite well. So in the meantime, we rely on our partners. Like I said, it's a collaborative project with the Forest Service and the Colorado Native Plant Society. So they're located all over the map. And so we might reach out before we go out to a forest, reach out to the forest botanist or range land manager and just ask them, hey, we are coming out to your forest in a couple of weeks, or we're thinking about it. We want to collect some grasses. How are the grasses looking on your forest unit? And even just at that very general level, they can usually provide some really good information. They'll say like, oh, well, I think it's going to be a little bit too early, or no, that sounds good. And they're usually right. So that's a really great thing about partnering with people in other locations, and it benefits us a lot. And then the Colorado Native Plant Society is also located all over, at least Colorado. You can see their different chapters on the map on the right. And they're also out on the public lands a lot, so we can also ask them. And we do ask them. And they also contribute through an iNaturalist project that they run, and we can gain information about the phenology of a given area through this iNaturalist project, as well as get some additional potential scouting locations through that. So, and speaking of iNaturalist, if you do have a species that is new to you and you don't know where to start or have no idea about its phenology, iNaturalist is a good place to start, because you can go to a species about page, and then if you click on this flowers and fruits tab, it'll switch this graph from just showing like when it was observed generally to when was it observed in these different phenological categories. And so you can look at the fruits and seeds category, for example, and see when plants in fruit or in seed were found. And you can even limit it to a specific geographic area. So like if you look up in the top right, I've limited it to just Colorado. And so that can be useful to figure out, just a baseline of where a species or when a species might be ready for seed collection in a given area generally. So yeah, that was my last slide. Thanks for listening, and we can turn it over to questions now if we want.
Yeah, thank you, um, Curtis. It's such a, we do a lot of seed collection. And it's so hard sometimes when like, every season can be really different. And so we love that you've attempted to create some sort of a model because it's something we've talked about so much. Like, wouldn't it be great? um, so yeah, I hope you keep sticking with it. Oh, good. We have a question.
Thank you.
Okay, so we have one. Is this model available publicly?
No, it's still a work in progress, as I alluded to in my talk. Um, so no, it's not. But if we when we do get to a point where there is something like shareable, like an app version of it or a interactive map, I'd be happy to share that with whoever's interested. Like I said, the data that we're using, at least the climate data, is publicly available though. So if you like we're using some like student internship, some students who are doing an internship with us. If you have somebody who has like some good coding ability, like I said, the data is publicly available, so they could maybe work with you too to develop something for your use case.
Okay, yeah, it looks like lots of people are interested. So, and we have a question from Alon saying GDD is really useful in agriculture. What references could you suggest for restorationists or collectors to read?
Oh, yeah. So that's actually, I don't have anything off the top of my head, but I can look for some. But that is one of the shortfalls of using GDD is it was developed for agriculture, and so it doesn't take into account effects of precipitation. So that's one of the shortcomings of the model. So I'm glad you brought that up because in agriculture, well, if you're irrigating at least, you don't have to worry about precipitation so much because it's pretty much constant and reliable.
Well, great. I don't see any other open questions right now. So if someone feels like they have a question for you later, they can go find your talk on the Hoover agenda. And we got one more. Okay, great. What's the best way to keep up to date with your work? If the model does become available publicly, where would we find it?
Oh, good question. I have my LinkedIn linked to my like Hoover account, so I would share it on there if you want to follow me on LinkedIn.
That's great to know. I'll be following you as well.
That's good.
Um, and then Andrew, do you have any information on seed cleaning?
No, because we send it to the Forest Service nursery and they do all the cleaning. Um, so yeah, we just send it to them totally unclean, and yeah, they deal with that. So unfortunately, no, I don't.
And then we have another one. Do you have the code available? And is it in R or Python or where?
Yeah, we can share. I can share some code. It's in R. It's R code.
Great. And I think we have time for at least one more question. So from Sarah, do you think germination is a factor that could be included in a future model? In my area, we're interested in fall versus spring germination of native forbs versus the invasives to help with invasive management.
Mhm. Yeah, I think it could be included in a model. This model, it would probably have to be a different model because the growing degree day, well, the growing degree day model has like a start date where you start accumulating growing degree days, which is usually like January 1st. So if you were interested in fall germination, you would either have to start like pick a new, um, you'd probably have to use a different model altogether because they're going to be initiating some growth before January 1st. So it wouldn't work well for like a winter annual or a perennial that, you know, I don't actually know if there are perennials that do that. It wouldn't take into account any fall growth, just starting in the like on January 1st.
Great. Thank you so much. I think we're going to move on to the next speaker, but appreciate your talk, Curtis.
Yeah,
Um,
sounds good.
yeah. And yeah, if you have more questions that didn't get answered, please put them in Curtis's Huva session where his talk is, and you can ask more questions. Okay, so next up, we have Mark Feely, and he is from Ernst Conservation Seeds. Mark, go ahead and start sharing your screen, and he's going to be talking to us about breaking seed dormancy.
Are you seeing my presentation on there?
Not yet.
Okay. Let's see. There we go. Should have it there, right?
Yes, I'm seeing your kind of your not the presentation point, but then like where we can see the next slide.
Let's see what happens here. I don't know if that's gonna. Oh shoot. That's gonna mess me up. Let me try that one more time, folks.
Yeah.
For everyone that is watching this, my presentation will be available upon request. You can send me an email, and that way you'll have it. And my computer seems like it wants to do a nasty thing to me here. What is it doing?
So no, but I could also work on pulling it up if needed.
Okay, did you see it a moment ago?
Yeah, we did.
Okay.
Yeah, display settings should have swap views option.
Is it showing now?
Yeah, can you do the swap view in display settings?
Yep, there we go. How's that?
Perfect. Thank you.
All right. Thanks for hanging with me, folks. All right. So the objectives of this presentation are to help growers to locate existing information about breaking seed dormancy and to share Ernst protocols for breaking seed dormancy for transplant production. Please don't get too wrapped up in thinking you have to write a bunch of notes here. If it's not on the screen, you might want to write it down. If it's on the screen, again, I'll share this with you. Ernst Conservation Seeds does not grow transplants of native plants for retail sale. However, for nearly 20 years, Ernst has grown 1 million transplants per year for production. On our own, we figured out how to break seed dormancy for approximately 200 species of native plants, just using the general insights one can get from basic horticulture textbooks on plant propagation. But I want to share some other places you can get some really good insights. And so one of those places is to join your state's nursery and landscape trade association. That is a really good place to look for information or to meet potential to meet peers, potential customers, and potential suppliers. Also, there's a conference called Cultivate 26. That's presented by American Hort. It's in Columbus, Ohio. Great training on growing transplants, including fertilizer or fertilization pest management. We're interested in seed dormancy because most native species that we've handled produce a combination of dormant and non-dormant seed, but often the percentage of ready to germinate non-dormant seed is insufficient to generate the transplants. The types of seed dormancy that we've encountered in the literature are things like hormonal, so it's a balance between two hormones that does not permit germination, an impervious seed coat, seeds that cannot, the seed cannot imbibe water because the seed coat just prevents that from happening. There can be a structural dormancy. An example of this is eastern gamma grass, a wild cousin of corn, has a structure that surrounds the seed that must rot before the seed can imbibe water. There's also immature embryo. Now, Ernst has not dealt with this in terms of germinating things, but it occurs on something I believe it's called black cohosh semicifuga racemosa. The seed has to go through a warm period during which the embryo matures, then through a cold, moist period to overcome the hormonal dormancy. And then there's the presence of germination inhibitors such as tannins. So where can you go to learn to break seed dormancy? Well, a really good starting point is Carol Baskin and Jerry Baskin's book on seed dormancy breaking. perhaps more important, and if there's one resource from this meeting, this presentation that I think is of immense value to you, it's Norman Dino's work. These are live links. If for some reason you click on them after getting the presentation from me and they don't work, I can send you the PDFs of those documents. He did a magnificent body of work examining seed dormancy across a really large slew of species. He may not have the one you're interested in, but he may have a close relative, and that'll get you a starting point. Michael Durr's done some great works on plant propagation of woodies. He worked on the book that's at the bottom there, and then there was Hartman and Keister's book that I got 40 years ago at Penn State, and that's what I used. The insights from there, just what do we do to break seed dormancy? All right, so let's look at the most common technique that Ernst has used with breaking seed dormancy, and that's cold wet stratification. So in a Ziploc baggie, we mix 3 to 5 times as much place in or vermiculite as seed. This mix is moistened with tap water and refrigerated. Don't freeze. Stratification occurs just above freezing up to a temperature, I believe it's supposedly around 50 degrees Fahrenheit. So no freezing. The length of stratification period required by a species depends in part on the origin of this. It depends on the species and the genetic origin of the ecotype that you're working with. So, for example, Ernst has grown Liatris spicata from Florida and Pennsylvania. The Pennsylvania ecotype needs 3 to 4 months cold wet stratification, whereas the Florida ecotype half that. If we have never germinated a species, we'll plant a test tray in the greenhouse, and with our ecotypes, we're going to bring test trays out 8, 12, and 16 weeks after planting. For most of our species, 4 weeks just isn't going to cut it. Ernst's second most frequently used dormancy breaking technique is gibberellic acid. In a Ziploc baggie, we mix the seed with 3 to 5 times as much place sand as seed. This mix is moistened with a 500 part per million solution of gibberellic acid, left at room temperature for 24 hours, or even refrigerated. We sealed the baggies in the early days when I was most deeply involved with that. We purchased gibberellic acid from our greenhouse supply firm as a concentrated solution called ProGib or Gib Grow. The seeds are then planted in the greenhouse. Now, for gibberellic acid to be effective, there have to be these microscopic pores in the seed coat that are large enough for gibberellic acid to pass through. We're not going to see those with our eyes or with anything other than an electron microscope. So basically, you're not going to see them. It's just a matter, you can test it. If it works, great, use it. Ernst has found this technique to be effective on some asters, Symphyotrichums, Eurybia, Billingeria genus. This is what I'm talking about there. Bidens, thin seeded caryxes, Ludwigia, Mimulus, Monarda, Enothera, Penstemon, Pycnanthemum, Retivita, Rudbeckia, Scirpus, Solidago, Vernonia, and Veronicastrum. It can be effective for some members of a genus and not others. So some asters, it's not very effective with. Others, it's very effective. Some caryxes, so the larger seeded ones, it doesn't tend to be effective. When testing this technique, plant a tray of untreated and treated seed in the greenhouse to confirm your stand is not just the result of non-dormant seed germinating. If the stand is equal between the untreated and treated seed, what you're doing is you're effectively selectively breeding the crop to have non-dormant seed. And we don't want to do anything that's selecting the species any more than we already are. Another technique, this is our third most used technique, is scarification. We use this on legumes. We also use it on a species called Ceanothus, New Jersey tea. We place a 220 grain sandpaper on a cookie sheet, place the seed on top of 220 grain sandpaper, and then attach an 80 grain sandpaper to a sanding block, and we scratch the seed between those. We use enough force to break the seed, about 1 to 2% of the seed. We add a rhizobial inoculant after that, and then we plant it in the greenhouse. Another way of doing the same thing is to take like a coffee can, if you had one of those old coffee cans, and you line it with 120 grain sandpaper and just shake real aggressively for 2 to 5 minutes. If you want to know what inoculant to use, you can go to this book by Allen and Allen on legumes, and you can find out what commercial form of inoculant is used. Some legumes don't need it. If you're growing a species for which inoculant may not be available, what you might want to do is when you're collecting your seed, take a soil probe, collect a little bit of soil from around the legume, and then you break that up and mix it with sand and put a little bit of that in the trays when you're planting to use as your own inoculant. A technique we use on larger seeded calyxes and members of the genus Schonoplectus, so these are wetland grasses, is what we call a cold float stratification. It leaches germination, inhibiting tannins from the seed while also stratifying the seed. We got this idea from collecting seed that was floating underneath the ice on a marsh in the winter and planting it and seeing how well it did. So we'll put the seed into a clean Ziploc bag or plastic bottle. We add tap water. It doesn't have to be anything fancy until the seed floats. We seal the Ziploc bag or cap the bottle and then place it in the refrigerator at 38 to 42 degrees Fahrenheit. If using a baggie, you might want to set it in a Rubbermaid or Tupperware tub or cut open a gallon milk jug just to protect against the baggie tipping over and the contents spilling out all over in your refrigerator. Each week, open the baggie or the bottle and slowly pour off the water. This is removing the tannins. After the water runs clear for 2 weeks, plant test plots or trays in the greenhouse every 2 weeks until germination occurs. Sometimes we'll follow the cold float technique with what we call hot water technique that I'll describe here in a moment. So the hot water technique, we use this on Ceanothus, New Jersey tea after scarifying the seed, but we'll place the seed into water that's been heated to 130 degrees Fahrenheit. We just let it soak in there. We don't maintain that heat. We leave it there for 30 minutes, and then we can pour off the water. Sometimes we'll follow this up with a 500 part per million 24 hours soak in 500 part per million gyrolic acid. With regards to certain native warm season grasses, our winters here in Northwest Pennsylvania are very overcast. We look at the sky and see what's that yellow object up there when we do see sun. We get germination of Indian grass and big blue stem when trays have been seeded for at least a week, followed by 3 clear days with greenhouse air temperatures reaching 90 to 100 degrees Fahrenheit. With some panicum species, Panicum anceps, which is also known as Coleotaenia anceps and Panicum virgatum, they'll benefit from a stratification I'll now describe. We'll soak the seeds for 24 to 48 hours in refrigerated water. We pour off the water. The seeds are then refrigerated for at 38 to 42 degrees for 2 to 6 weeks. If given the 2-week treatment, they need to be planted within 6 to 8 hours. I say that the real hard, fast figure is 12 hours. Seed dormancy starts returning after that. If given a 6-month or 6-week treatment, the seed may be dried afterwards and planted anytime within 6 to 12 months. You got to get it planted within that 12 months or the viability is shot. Treated seeds using this technique will germinate about 7 days after planting with full germination after 14 days. In contrast, without this treatment, it's about 28 days to get full germination. When you're storing the seeds that have been treated with the 6-week treatment, you want to keep it refrigerated and cool during that period. The viability, again, will crash on this within 12 months. With regards to little blue stem, Schizachyrium scoparium, we've only had success by planting in late March or later. We think this is because there's a phytochrome response, a light wavelength that the seeds picking up. And again, we're very cloudy in winter months. So, and we found artificial light and root zone heating had no impact on earlier plantings. We've also found that we're best off planting milkweeds, baptisias, and lespedezas in March or later. They germinate earlier, but they just have issues and have done poorly for us when we've planted them in December, January, February. For greenhouse planting, we often we typically hand plant our seeds. We'll mix our seeds with place sand brand sand to dilute it to 3 to 7 seeds per pinch. We plant 1 pinch of seed sand mixed per cell. Actually, I'm wrong. 3 seeds might be something like an iris. 7 seeds is more typical. Some it's going to be up around 10. Our growth media is a peat-based plug mix. We avoid bark-based growing media because we had issues with that in the past. Sometimes we've amended the plug mix with 1/4 volume of perlite, fine grain rice hulls, or rock wool. When seeds are germinating, the cotyledons are the first leaves of a plant, and they were wrapped up inside the seed. Cotyledons of dicots typically resemble the shape of the seed, whereas cotyledons of grasses and sedges and rushes often resemble corn seedlings, but they may be smaller in diameter, more compact, or more leggy. If the potting soil or crop seed is contaminated with weeds, the weed seeds typically germinate earlier and appear different from most of your seedlings. Unless potting soil has been sterilized through steam treatment, it's likely to have spores of fungal seedling diseases called Pythium or Phytophthora. These fungi often manifest themselves by the 2nd to 4th week after seedling emergence. In the case of Phytophthora, in some Pythiums, you can get a vaporization of seedlings in 24 hours. If you think this might be occurring, take a picture of a set of trays each day with your cell phone, and if you notice it's thinning, you need to do a treatment. We found metalaxyl in the form of Subdue to be a very effective control. You'd be wise to consult with your local cooperative extension specialist about common insect and disease pests observed in your area, and these should be monitored and treated using IPM. Some other final thoughts here. We grow our transplants in flats with 128 cells that are 1 inch by 1 inch by 2 1/4 inches. We aim for a soil pH of 6.5 to 7. And about 1 month after planting trays in the greenhouse, we begin applying fertilizer. For fertilizer, we use 150 parts per million of 1555 until the plants are 1 to 2 inches tall. At that point, we switch to 150 parts per million, 20-10-20. When planting follows dormancy breaking, germination typically occurs in 7 to 21 days, as long as our greenhouse temperatures are reasonably warm. From germination to root balled and ready for transplanting is typically 3 to 4 months. Some species produce rosettes that are tight to the top of the tray and thus difficult to remove during transplanting. To get the stems to elongate, we spray the plants with a 5 part per million solution of gibberellic acid. And with that, you might want to take a quick photo of what's there so that you can see my contact information. Against mark@ernstseed.com. mark@ernstseed.com. And I'm going to take that off there. All right. And stopping the sharing.
Perfect. Thank you so much, Mark. I think we have time for at least one question. So I'll start with the most upvoted one. And it's, can you speak more about the need to make sure the method that you are trying results in a majority of seeds germinating in order to avoid inadvertent selection in dormancy traits? I think some folks think that even a low or moderate germ percent can be a win for hard to germ species, but that still leaves out the ungerminated seeds genetics.
Yes, and you are selecting the plants for lack of dormancy if you're not getting a significant dormancy breaking. The date we collect the seed from the foundation population, where we collect our selection events, and our germination can be a selection event. If we're trying to maintain diversity, it's good to have the best germination as possible. So that's why we're aiming, and for practical purposes, when Ernst is growing transplants for production fields, we don't want to have to grow 1,000 cells to get 500 seedlings. We want to be able to grow 500 cells to get 500 seedlings. And we want those cells to be root balled fast. And so if we have 3 to 5 seedlings per cell, they're root balled within 3 months.
Great, thank you. I'm not sure we'll have time to answer any of the others, but you had a ton of questions, Mark.
So if I could throw a couple last things out, I am doing a presentation on small scale seed collection, drying, processing, and storage. That's going to be offered through National Cooperative Extension, and I'll try to share something about that on LinkedIn. I don't know exactly when it's going to be, but just wanted to let you all know that. If you email me, I'll try to send you back something when I hear the date or how to log on to it. Okay. And it is free.
Oh, that's great. Thank you, Mark. And you can also connect with Mark on Whova if you missed his email. or find his email on there as well. And then also can ask some more questions within his HUVA agenda session. Thank you so much, Mark.
You're welcome.
All right. Oh, sorry, I muted myself. Next up, we have Alliy Gundlach from Tree Pittsburgh, and she will be talking about growing trees from seed, small scale nursery making a large local impact. So you should be able to share your screen now, Alliy.
Okay. There we go. All right. Does that look okay?
I think also we need to switch to presenter view.
Oh, let's see. There we go. How's that?
Perfect. Thank you.
Okay. Yeah, of course. All right, I'll go back one slide here. Well, thanks so much, Morgan. I'm happy to be here and just so inspired by everyone who's spoken before me. But like she said, I'm just going to be talking a little bit about our seed program here at Tree Pittsburgh, what that looks like, and how we really are making an impact within our community here in Southwest Pennsylvania. So first off, we'll talk a little bit about what Tree Pittsburgh is. So we were founded in 2006. So it's our 20-year celebration this year, which is pretty cool. We're an environmental nonprofit dedicated to strengthening and building community vitality by restoring and protecting the urban forest through tree planting and care, education, advocacy, land conservation, everything that's listed there. And you can see within the programs that are under the umbrella of Tree Pittsburgh, we do a lot of different things. But one of those programs specifically is the Heritage Nursery, which I work for. I'll talk a little bit about the nursery here. So you can see here, we grow over 110 species of diverse regionally adapted trees and shrubs grown from all locally collected seed. So our main focuses within the nursery, obviously, are genetic diversity. We want to have long-term survival, growth, and health of all the trees that we're planting out. We want to be locally adapted, hence the seed collection within our ecoregion. And we also focus a lot on quality root systems. So we're applying horticultural practices that support healthy root development, because then we get, better survival rates out in the field. We're located, I think you might have seen in the very first picture, on a 2-acre area within the city. So we are an urban nursery, and we grow about 50,000 seedlings per year and turn that over. So those go into different restoration plantings, whether it's within our own Tree Pittsburgh plantings, or if any of our partners, we partner with a lot of different land trusts, foundations, county parks, city parks, game lands, all sorts of places use our nursery stock. So I've heard one of our educators always says that we're growing the future forest of Southwestern Pennsylvania, and that's what we aim for. So what under the Heritage Nursery is my program, the Seed Program. So I'm just going to talk about the 3 main silos that I focus on, collection, processing, and propagation, and also how we engage the community through those 3 things. So really the who and why of our collection, like I mentioned before, we grow about 110 native species, but we're always open to requests from the restoration community. So some folks may want something that is rare that we may not grow regularly, and we can do that for them. We are wholesale, and so we don't have a retail open, which is actually much easier for our nursery staff. But we do have a lot of selection guidelines about what we are growing. So we always want to consider our urban setting, even though we do have partners that purchase from us from outside of the city. We're always thinking about what is tolerant of heat and drought and salt and pollution, because we really want to be growing the best nursery restoration stock we can for our settings. Also, we always want to consider native ranges of things. It was founded originally in 2018, the nursery was, because there was a lack of good restoration stock for our region specifically. And at the time, some just tree enthusiasts started growing all sorts of different trees, some of those being what would be considered like assisted migration, more southern species, And we've pretty much nixed those from what we grow now. So this is all kind of like learning and evolving over time. We always want to make sure, obviously, nothing on the invasive list, but we also think now about aggressiveness of those species. So if, it might influence a planting site. So that's something we take into consideration with our selection guidelines. Also, how available is it in the field? How accessible is it? And what kind of benefits is it bringing? What kind of biodiversity are these species supporting? And we let those also influence what we're growing. So where we collect, and you can see here where Pittsburgh's highlighted or circled in red there, that's our ecoregion, the Western Allegheny Plateau. So every winter, it's a whole process of getting permits and permissions with all of our different partners. Like I mentioned, pretty much everyone who purchases trees from us, we also have partnerships with to go and collect seed on their land. We have a lot of different partners that we work with, and we're lucky that way, who really support our mission and what we're doing. And we've developed a lot of protocols, and I share these out every year just as a reminder, even if I talk to these folks on a weekly basis, that reflect our values. And I'll go into those a little bit more. And we've expanded our collection recently. Typically, we did it within, the county that Pittsburgh sits in and maybe a few counties south of us. But now I do pretty much a biannual trip into southeastern Ohio and into the panhandle of West Virginia in order to support resilient stock. So we have a lot of different protocols now about how we collect. And these have developed over time. So mostly they're collected by hand. I use pole pruners, tarps. The root apron is always really helpful. I use the acorn weasel occasionally, but it also picks up deer poop, I found out. And we have pretty high deer pressure here, so I don't always love sorting through that. So I don't use that a ton, but if you don't have deer pressure, I would recommend it. But another protocol we always follow is that we try not to collect more than 10% of any one specimen's yield. That's a really conservative percentage, but because we don't grow a ton, we have that luxury to be really choosy about what we're using, and also not to disrupt any of the ecosystem benefits. We always prioritize collection from many of one of a species rather than a singleton, and we don't collect from the same tree year upon year. We try to collect from 5 locations, at least 1 mile apart. Usually it's much more than that for each species that we grow. And also mentioned here, and like in the picture here, we have a lot of really great partnerships. Arborists like this one here was donating their time and their bucket truck in order to do white pine seed collection because, can't climb those, they're pretty unstable, and all the good seeds up at the top. So since they're wind pollinated. We also partner with city forestry and park rangers a lot. So the parks that we do partner with, I'll do annual seed collection classes for their rangers. And whether they use it and donate the seed or not, it's still just sort of like a fun educational thing that we can give to them for use of their land and seed. And and how we use this to engage with the community is 2 different ways. We do a lot of classes throughout the year and also a lot of volunteer work. So we do a seed collection class every August. So it's just talking about the basics, the different techniques, how to tell if a seed is ripe. We really highlight the ethics of seed collection, so there's not this over collection. And it's actually been really cool to watch 2 different individuals create their own hobby nurseries, native tree nurseries. And they sell them at farmers markets and stuff. This is what we want to do. We want to share our knowledge with folks, and the more plants we can get out there, the better. I also created the Seed Seekers program, which is a volunteer program. Last year was the pilot year. It was in partnership with our city forestry, where they gave me permits to allow 6 different individuals who got assigned to parks close to where they live to go and check on those parks for 3 months during the height of collection for us, which is about August to the end of October. And because of this program, it was really awesome. We had over 70 donations, 70 distinct donations with 42 hours of volunteer time. So we're going to be repeating that this year, possibly including more parks and more individuals. So moving on to the processing and stratification silo of the seed program. So like one of the presenters shared earlier, thank you. We've also created a seed Bible. So this is information that we've taken from all sorts of different resources, but then also all of the knowledge that we've gained year upon year, experimenting with different things. So whether it's stratification times or cleaning processes or sowing, how to set up to do all the sowing for these species. It's really nice because you can see I have two of the nursery technicians who were here last year, and I could ask them, hey, could you clean the elderberry? They flip open the seed Bible and they have all the information they need there in order to do that. So that's been a really cool resource, and we update it yearly with any new learnings. And also the seed bank has been really important for us. This is something that has started small and is developed over time, where, the nursery director maybe gives me the projected numbers that we want to grow for the next year. I take that in consideration into what I'm putting into stratification, along with germination rates and mortality rates. And then I save the leftover seed. And so there's been so many good benefits to this. So it's, lessens waste in the nursery, lessens seed waste, decreases nursery pressure because there's nothing like having too many seedlings and feeling like, what do I do with all of these? And it's also avoided gaps in production, so that natural periodicity that a lot of trees and shrubs have. We fill in those gaps with seeds that we've saved from previous years. And it's been a really cool resource for the community. We've had folks reach out who were doing like seed balls, for example, or seed bombs. And, they're doing them along this sort of like wetland area. So I could give them a bunch of buttonbush seed or silky dogwood seed that they could mix in to those seed balls. So that was kind of a cool partnership and things that we have that we just give away in our community. With that similar vein, there's always engagement across everything we do. So the fall sewing classes, the goal is always to demystify and to help folks feel more comfortable with growing their own stuff. And the weekly volunteer events as well throughout the month of September. And it's just teaching people how to clean, and we get so much done. And a lot along the same lines as sowing all the recalcitrant seeds, so acorns, buckeyes, and hickories that we do in the fall. Last but not least, propagation. So all the sowing in the spring and managing the greenhouse is done. So pretty much a pretty basic growing setup with, always making sure things are sterilized. We use Xeritol to dip all of our trays, but pretty much all of our sowing is done within a six week period. You can see from the picture there, we use mostly these 15 by 15 boxes. Mostly because we can fit more of them in there. We are outfitted with one greenhouse. And in our 2-acre growing fields, we do have 8 hoop houses along with regular growing rows and a pole barn. But just for space sake, we can start more in these before they get potted up either later that year or the following year. We use 5-inch tree pots for certain things. I mentioned root health, so things with tap roots we often do in tree pots, and containers for our conifer species. So a lot of things that, a typical greenhouse, what we like to use is the ProMix Super Sacks. We were getting them in individual bags, so it was a better deal somehow, but we just hated the plastic waste, so got rid of that. We found that pre-moistening the soil was really important for germination rates, because sometimes that soil just takes forever to wet up. And mixing in amendments for certain species. This is also something I would note in the seed Bible about adding more perlite for drainage or peat for acidity, and just making those extra little tweaks for certain species that may be tricky. And last but not least, the propagation engagement. So weekly seed sowing, we have a whole group of folks, and actually kind of like a cool little wait list of extra people who always want to come on and help. It's a really accessible form of volunteering because people can sit down, oftentimes in the nursery where people want to help. It's moving heavy things, it's moving pots, dragging carts around, things like that. So this is really great to get anybody who might be interested out volunteering. And also we have 3 classes throughout the year. So the spring emergence class, the collection class and the fall sowing class. So that's the end there. So yeah, thanks for have any questions.
Great. Thank you so much, Alliy. It does look like we have some questions. And so I'll just start with the top most upvoted one. Your seed Bible sounds wonderful. Is that a resource that you're willing to share?
So yes, we do have future plans to actually put it into production at some point. We'd love to share that information. If anybody has questions about certain species, I'd be more than happy to like help them or hand over the information that we do have right now. I want to collect more data before we fully make it official, though.
Great, thank you. And then from Amy, do you collect any recalcitrant seed, or are you mostly collecting orthodox seed? If you do collect recalcitrant seed, do you store it for any period of time?
No, we don't store it. We sow everything immediately. So, I mean, the thing that comes to mind is always like the pawpaws, the oaks, acorns, hickories, maples. Like none of that stuff really keeps. I mean, maples we've stored for like 2 years at most, but most of that stuff isn't going to keep. So we just sow immediately.
Yeah, that makes sense. And from Asa, for the trees without tap roots, are you removing them from these 15 by 15 trays as bare root and potting them directly into gallons?
Yes, that's exactly right. We actually pot them up into 2 gallons the following year. That seems to be the best size restoration stock that we have really good. Like our field crew gives us feedback, and that seems to be good for roots, but also for survivability because our soil is so degraded here often that a more robust root system is really helpful.
Gotcha. Okay, another question about your seed Bible. um, so I'll say you answered that for now. And then from Rachel, thank you for all you shared. Can you say more about how you grow your trees for good root development?
Sure. Yeah. So we use a couple different techniques. Like I mentioned, the 2 gallons, and within that up potting process, we found that it's really important to really spread out the root like really spread out those roots because oftentimes we started doing a lot more education with volunteers because the roots would just get kind of shoved down in and pounded down. So we take a lot of care whenever we're transplanting. Also, we do a lot of different types of air pruning. So having things either up on benches or some type of air along the bottom. We also have specific pots that once the root hits the air, it won't grow there. So a lot of air pruning techniques. Yeah, it's just making space for the taproot. Yeah, that kind of stuff.
That's great. I think
Hmm.
we still have a little more time. So
Okay.
from Russ, it looks like you're using peat in your growing medium. This is a concern for some of us because of the adverse impacts of peat mining. So some of us are trying out non-peat alternatives. There is peat-free, a Pittsburgh product called pit moss. Have you tried
Hmm.
using that?
Yes, we have tried using it, and I could see it being a great product for like if somebody had container gardens or something like that. We found on large scale that it dries out and has a really, really hard time wetting back up. Um, and as much as we want to support that product, and it has definitely its place and use for large, like our scale production, it didn't quite fit the needs that we have. But yeah, that's a really good question.
Yeah, it's a hard one.
Mhm.
Thank you. Yeah, and then someone did comment that mushroom compost works great for them too. Um,
Mm,
I think we are close. I think we'll just move all the rest of the questions to Allie's session in the agenda. There's a few more questions in here. People are very interested, and we enjoyed you sharing your work. So yeah, you can find Alliy on her agenda session and ask the further remaining questions there. And then with that, I think we'll stop sharing and add in the next speaker. So
sure.
thank
Sounds
you,
good.
Alliy.
Thanks. Yeah.
Okay. Okay, great. So next we have Rob Massatti from Landscape Stewardship Collective, and he's going to be talking about maintaining genetic diversity during the agricultural increase of native seeds. So, yeah, we'll let you share and take it away, Rob.
Thank you. I am worried about my connection, so I think that I am going to stop my video for now. I will come back on after the end. All right, so hopefully, can you hear me and see this okay?
Yes, this is good.
Perfect. Okay, it is really a pleasure to be able to talk to you all today, and I've really been impressed by how diverse a group is attending the Seed Conference this year. So kudos to you all for the great work that you're doing. Today, I want to talk about something that it sits directly at the intersection of science and practice, namely, when we take wild collected seed and increase it for use in restoration efforts, what happens to the genetics? Oh no. Okay. But before anything else, I'd like to acknowledge my fantastic collaborators without whom this work wouldn't have been possible. Trevor Fask did the majority of the work that I'll be presenting, and this larger group has been looking into the grass species used here in lab and field-based experiments. Some of these folks have moved on from these places that they're associated with here. This project has been going on for quite a while now. Okay, so I'd like to set the stage for you all and place this study in a broader context, the broader context of restoration. So starting really, really big, we all know that restoration is inherently difficult. We work in landscapes shaped by disturbance, invasive species, altered fire regimes, and increasingly unpredictable climates. And we faced numerous procedural challenges, such as how we do our restoration, what we use for our restoration, how we account for changing climates. A lot of what my research focuses on is the what of restoration, so the species that we use and the intraspecific sources that we use, both in terms of their use and their production. So at the same time as we have all of these challenges, disturbances are incredibly common across our landscapes, and management actions take significant resources. This figure shows an exercise that we did characterizing different categories of disturbance across the Colorado Plateau. For example, grazing, wildfire, and energy. And it's evident that when you add all of these up, such as on the right here in yellow, a significant portion of the plateau may benefit from some type of management action. And this is reiterated across the West and North America. How we successfully conduct landscape management involves many evolutionary and ecological questions, but to make seed available at scale, we quickly have to address agricultural questions as well. And thankfully, we have a framework which you are all familiar with to increase the diversity of species and sources available for land management. The seed strategy gives us a clear vision for using genetically appropriate seed in terms of the right seed in the right place at the right time. To meet that outstanding restoration demand, we almost always have to collect seed from wild populations and put it into agricultural production fields. And there are implicit concerns that agricultural increase may alter genetic composition in ways that reduce genetic appropriateness. And so today, that is where we are spending this next few minutes. So the study that we did that was funded by New Mexico BLM, Plant Conservation and Restoration Program, as well as USDA AFRI. This project used 3 different workhorse restoration species, common across the West and used in dryland restoration in many, many areas. Bouteloua grassless, Bouteloua curtipendula, and Sporobolus cryptandrus. So these species are known to have life history characteristics that may cause problems for seed increase. The two grama grasses have both polyploidy and aneuploidy and some weirdness when it comes to their mating systems. Sporobolus cryptandrus is self-fertilizing. So, so we really need to understand how these life history characteristics are interacting with production. Seed was collected from 10 to 15 wild populations according to Seeds of Success protocols by the Institute of Applied Ecology in the New Mexico BLM. And seed sites for collection were determined by selecting those with environmental similarity. I can answer more questions about that at some other time if you are interested. At the same time as seeds were collected in the field, leaf tissue samples were taken from plants in the wild, and those were used to create a genetic data set that we'll call wild. And the collected seed is G0 seed according to AOSCA guidelines. So the seeds that were collected within the field were used to plant two different types of establish two different types of fields. The seed sources were kept separately and planted into rows so that we could track identity of the seeds. And we have genetic data from those. But then also the seed was pooled and used to establish a larger production field. And this was what was harvested for G1 seed. And we have genetic data from the pooled field as well, and the pooling is much more common in terms of seed increase compared to keeping the sources separate. So it's important to note that all fields were established from greenhouse-grown transplants, and this will come up a little later. And finally, the G1 seed was harvested from the G0 pooled field, and that led to a G1 genetic data set. So we have 4 data sets per species for each of 3 species, and we are looking at how genetics are shifting across these data sets. So we ended up with genetic data from over 500 plants per species and thousands of genetic markers. So we had quite deep data that were very informative. And we're just going to jump into results. So I'm going to be showing you this figure multiple times. What you're seeing here is data for the 3 species, which are in columns with blue grama grass on the left, side oats in the middle, and stand drop seed on the right. On the top are illustrations that illustrate genetic diversity. And on the bottom, we're looking at genetic differentiation. Okay, so if we zoom into genetic diversity, what you're seeing here is diversity values and their means, which are the clear symbols across the wild G0 and G1 generations. And you can see that there are differences, and we'll look more in depth at these. But for Gracilis on the left, you see that the 3 generations are more or less equal. In the middle, for Curtipendula, you see that there's an increase in diversity in G0 and G1. And then for Sporobolus on the right, you see a decrease in G0 and G1. So for differentiation, this is how similar basically the gene pools are between these different pools of individuals. Ultimately, or optimally, we want to see zeros in those clear rectangles between the colored shapes. Zeros would mean that there is no difference in the genetics between the different pools. Whereas as you get closer to 1 means you have completely different gene pools. So we see different values of differentiation between the species. So obviously our question is what's driving differences among these species? And we're going to look at the species one at a time, first with Bouteloua gracilis. And I'm showing you now the same figure as we were just looking at. And as a reminder, we see fairly consistent genetic diversity on the top and then pretty low differentiation on the bottom. And I'm going to show you one other type of data graph. I'm sorry I don't have more pretty pictures. I feel bad because everyone else has such nice pictures. Um, so this other type of graph I'm going to be showing you for each of the three species is a principal components analysis, which you really don't need to understand. What you need to know about this graph is that each one of those symbols is the genetic data from one individual plant. So there's a lot of different symbols there for each individual. So in this figure, they're colored by the source population, so by collection location, and then the circles represent individuals that were sampled from the wild populations, and the squares were sampled from the agricultural field. And so, for example, on the bottom left, you see lavender symbols, and you see both circles and squares of the lavender. So this is exactly what we want to see. We want to see individuals in ag fields looking like the wild individuals. And if you look across the mass of points there, you can see that pattern is reiterated across the different source populations. And this is really what we expect for an outcrossing species. And despite the fact that I said that Bootaloo agracilis has a lot of ploidy variation, most of the sampling sites that we collected from were tetraploids. So this really represents more or less all tetraploids outcrossing. These patterns actually look really pretty and nice. And this makes a lot of sense in terms of the figure on the left, where we see consistent genetic diversity from wild to G1, and then low genetic variation. All right. So next, we're going to look at the Bootaloo critipendula. As a reminder, we saw increased genetic diversity in G0 and G1 and increased differentiation between wild in G0 and G1. Again, we're looking at a PCA now, but this time the colors do not represent sampling locations. They represent genetic clusters. So the fact that all of these points are clustering like into these 5 different clusters indicates that we have biological processes above and beyond just normal outcrossing. And so what those really relate to, first and foremost, is ploidy. So individuals that are dark blue are diploids, whereas the other clusters correspond to tetraploids. What's really interesting about this is that if you look between the circles and the squares, especially with the diploids, you see a significant drop-off in the ag field of diploids. So what we're seeing here is that tetraploids become overrepresented in the agricultural fields, which drives higher genetic diversity. And I can go into that in more detail if anyone is interested at another time, as well as higher genetic differentiation. So overrepresented tetraploids. And finally, we have to look at Spiralis cryptandrus, which is our selfing species, and where we saw lower genetic diversity in G0 and G1 and higher differentiation. And finally, here's our PCA from that. Again, we see these really noticeable clusters of points, which indicates, in this case, we're looking at evolutionary lineages that have been created through time of selfing individuals. So there's not really crossing between these lineages. And what's really interesting is that you can find all of these lineages at one sampling site. So these different genotypes are all co-occurring, and yet these individuals aren't interacting with one another at all. And to bring this back to the production field, in the production field, we basically see only one of the genetic lineages represented, and which is the reason we are seeing this incredibly low genetic diversity in G0 and G1 and high genetic differentiation. So here are patterns, and let's see here, I'm pushing up on my time already. But one thing I want to note is that the actual process of agricultural increase is not the culprit. So look at the values in the rectangles between G0 and G1. They're all close to 0, meaning that the G1 seed represents the G0 individuals really, really well. And so what are we actually talking about here? If we created a hypothesis for our selfing species, we would have expected on the left that we collected seeds across our wild sources. And then if we establish our field from the G0 seed in the middle, we represent all of our wild sources in the field. And because they don't cross in G1 on the right, we see all of those genotypes. But this is actually what we saw. We saw that our source populations had all of the different genetic lineages, but that only one of those lineages was established in our production field, and the G1 seed, of course, reflected that. So, and this is the same as what we discovered for the polyploid Bouteloua curtipendula as well. But what this really indicates is that the genetic shifts are occurring between seed collection and field establishment, not in the increase itself. And so our data aren't really appropriate for understanding how those shifts could have occurred, but there are tons of hypotheses that we can come up with, including that certain genotypes flowered and set seed at different times of the growing season, or even in different years, or that seed storage or greenhouse propagation favored one genotype over another, or one ploidy level over another. So really, we know something very fundamental about the increase process now, but we have more questions about what was actually driving the process. So finally, our take-home messages in our quest to increase the number of species and sources available to conduct ecologically and genetically appropriate restoration, species biology really, really matters. For outcrossing species, we may have fewer concerns, but as soon as you get species that have ploidy variation or different types of mating systems, we can run into significant problems if we want our native plant materials to accurately represent wild populations. And we really need to design more targeted experiments to understand what about the seed collection and establishment our practices are actually causing these shifts. And I think an outstanding question is, does this matter? Do we need all of the genetic lineages represented or ploidy levels represented? The fact that they are represented in natural populations does indicate that they should have some importance, but we don't really understand what it is. And I'll just say that we've seen similar patterns in Indian ricegrass, Achnathrem hymenoides, and other species. So this isn't confined to just a few species, but it might be more ubiquitous than we actually understand. And I'll just put in a plug here. If you are like one of the big holes here is that we don't really have enough tracking of how seed sources are used in restoration projects across environmental space. This is a huge knowledge gap that has been identified by federal groups as well as private sector collaborators. And I think Laura Shriver will be talking about this more tomorrow in her talk. But one thing, we need better data about seed usage. And if you want to share data, I would be happy to receive it and use that data. Oops. Okay. And finally, this is my thank you. I really appreciate all of your attention. And yeah, thank you.
Great. Thank you, Rob. Maybe we'll try one question. How do you view ploidy testing as being a part of the accession building process?
In my opinion, if you know you have a species with ploidy variation, you need to be characterizing ploidy of your wild sources before you take them into production. the problem is if you mix, for example, if you mix a diploid and a tetraploid population, you might expect that you get diploids and tetraploids in your G0 production field, but when those cross and you get your G1 seed, you could be coming up with a bunch of triploid seeds that either are inviable or have, lower competitive ability. And so ploidy is something especially that you need to be very aware of.
Well, that's great. Thank you. There were a lot of interested questions and comments, so please go ahead and put them in Rob's session. I'm sure he'd be happy to answer more questions because there's a lot of interest in this. So thank you, Rob. Okay, next we're going to go into our lightning sessions. And so I'm going to share my screen and I'm going to advance this forward for these folks. So let me do that and then I'll introduce our next speaker. All right. You'd think. Okay. So next we have, sorry, I'm getting all my stuff together. But next we have Alissa Kim from the University of Nevada, Reno, to also talk to us about some farming of native plants. So let me get this in presenter mode. All right. Go for it, Alissa.
Hi, thanks so much for the introduction and thanks for having me here. My name is Alissa Kim and I am a graduate student with Dr. Beth Ledger at the University of Nevada, Reno. And today I'm going to be talking about farming for native plants, a very good segue, a comparison of seeds and seedlings of Elymus next. So starting off, the Great Basin is a lot of seed-based restoration. It's a large cold desert with high disturbance history. So we're looking in the area of Nevada, California, Utah, Idaho, that area of the United States. And we annually seed about 300 to 350 hectares. You can see on the right side that with more current years, we have increased that seeding. And because of that, seed needs are much greater than the current available seed. So one solution for that is to use farming practices to produce native seed. Next. Agricultural increase can have benefits over wild collecting. Some of those benefits can include increased yields, more efficient harvests. You can conserve wild populations while still using localized seed, and it is arguably less labor and time intensive. Next. However, it's not a silver bullet. So some of the concerns around agricultural increase include reduced genetic variability, inbreeding depression, reduced diversity of species used. It can be expensive. And then the last point, which we'll really hone on today, is accidental selection or undesirable maternal effects. Next. So my overall question is, does agricultural increase of native seed change phenotypic traits for Elymus sp? And you might have remembered that I had elymoides and multisetus on my title. For this presentation, we're just going to treat them as one and the same. They're pretty taxonomically similar and they do co-occur. So Elymus sp just for now. Next. So why do we choose these populations to test? There was a previous graduate student, Allison Agnery, you can see her face right there, who went out and did all of the grunt work of looking at all of these different populations. And we chose 3 populations of Elymus sp and 3 populations of Achnotharum thurberrianum. These seeds were wild collected in Nevada and California, and they were chosen based on high survival rates in burned and high cheatgrass invaded areas. And then these wild collected seeds were given to a farm to BFI native seed. Thank you so much, Matt and Jerry Benson, in a wonderful state of Washington. And they did all the hard work of growing up plugs and then transplanting them in 2022. In 2023, which was the first year of harvest, we saw some really high yields. However, in 2024, which was the second year of harvest, we saw much lower yields. Next. So here are some of the phenotypic traits that I tested for. The first one was seed mass. I weighed 1,800 seeds, 200 seeds per population and harvest. And then germination percentage was looked at by taking each of those individual seeds and putting them into their own pots. And I tracked their population and harvest across this process as well. Emergence time was also tracked. I looked 3 times a day at 7 a.m., noon, and 8 p.m. to see when they emerged. And then root mass allocation, we harvested those plants and took them apart from roots and shoots and then scanned the roots and then dried them separately and weighed them. And then whatever plants we didn't harvest, we also put under a drought tolerance treatment. So half of those were then split again between control and drought. And the drought saw an increase in days between watering, and we weighed the dried shoot and root mass for those as well. Next. Today, though, we're only looking at the first 3. So seed mass, germination percentage, and emergence time, and kind of ignoring those latter. Next. Why were these traits chosen? So there was a paper published in 2019 by Dr. Beth Ledger that modeled a lot of different phenotypic traits for survival, for best survival in the Great Basin. And these are some of the traits that most stood out. You can see on the right side, higher seed mass, lower emergence time, and lower root mass ratio were good predictors for higher survival rates. And these histograms show all the seeds that were planted at 5 different sites, and the darker colors are the seeds that survived. Next. So again, my overall question is, does agricultural increase of native seed change phenotypic traits of Elymus sp? Next. And spoiler, they did change a little bit. They were pretty population specific and largely tended to be positive. Next. So we're going to look at seed mass first. Across the board, seed mass increased with cultivation. Whether that took a first harvest or a second harvest depended, but they did all increase their seed. So you can see harvest and population on the x-axis and seed weight on the y-axis. Like we said before, higher seed mass is a good predictor for higher survival rate as well. Next. Germination percentage differed a lot by population. We did see for some populations, they did have a small decrease in their germination. However, one of the populations had a pretty low percentage of germination for the wild collected seed and then doubled with cultivation and increased even more after that. So really depended based on the population. Next. And then lastly, emergence times decreased slightly. You can see that hours to emergence did get a little bit faster after the first harvest. Whether they stayed as fast or kind of reverted back to their wild sourced time depends for the second harvest, but they did get a little bit faster. Next. So overall, did they change? Yes, they did change, but overall in a positive direction. Negative changes were pretty minimal after 2 years of harvest, about a 2 to 16 decrease in germination percentage, and populations really differed in how they changed. Next. Lastly, I'm going to be looking at that root mass allocation and the drought tolerance information, and then also looking at the seed mass for Achnitherium thurberrianum. There's also genetic sequencing being done, not by me, but by Seth Romero, who is in another lab. And he is looking at the genetics for the seeds of from these populations and from these harvests as well. Next. Lastly, this research has strong implications for seed-based restoration in that using local ecotypes for restoration through agricultural increase can preserve locally adapted traits. There was no evidence in my research to show genetic drift over these short production periods. And ultimately, agricultural increase may be beneficial for seed-based restoration efforts when using local seed and over just a few harvests. Next. With that, I want to say thank you so much to my advisor, Dr. Beth Ledger, my committee, my lab members, all the undergrads who have helped with this project. A huge shout out to BFI Native Seeds again, and to the greenhouse staff. Next. And with that, I can take any questions, or you can also email me if you have any follow-ups.
That was awesome. You did so great, Alissa.
Thank you.
Let me do one question for you. So nice work, Alissa. Are you planning on obtaining any genetic data to compare to your phenotypic data?
Yes. So I am not planning on doing that, but there is a PhD student. His name is Seth Romero. He is in Tom Parchman's lab, which is also at the University of Nevada, Reno. And he is going to be doing that work in tandem with other genetic work. But he has run all of the genetics for both the Elymus and the Agniferum seed. So yes, there will be a genetic component to the phenotypic trait component as well.
Awesome. Thank you so much. We really appreciate it. And yeah,
Thank
like
you.
said, you can reach out to her anytime, them anytime. All right. On to the next. We have, sorry, my stuff is all okay. Yes, we have Laura Fischer-Walter to talk about from the UNI Tallgrass Prairie Center to talk about experiences with 2 perennial hemiparasites in agronomic seed production. So let me get some things sorted and take it away, Laura.
Okay, it's kind of funny to be following in the footsteps of folks who are providing lots of wonderful data slides, and I'm going to be talking strictly from an experiential perspective here, working with 2 species of perennial hemiparasites in the genus Pedicularis and attempting to grow those out for, pardon me, for seed increase. Next, please. I am located at the University of Northern Iowa in northeastern Iowa at the Tallgrass Prairie Center, where we have 4 programs that interact with each other, all of which help to empower people to restore prairies in various contexts around our state and region. And I happen to manage the plant materials program. Next. In this program, we do make wild collections, and then we increase that seed in seed production fields located at the UNI campus. And we make the seed that we produce then available to native seed growers in the commercial native seed market who can then produce it at a large scale and are eligible for source identified certification for that seed. And we hope to make a diverse and regionally appropriate seed available and affordable. Next. So one of the things we do is we look at what's available in the native seed market from purchases made by our other programs at the Tallgrass Prairie Center, and we look for gaps. And one of those that we noticed was these perennial hemiparasites that were poorly available as Iowa source identified seed. We think that they are likely to be suitable for roadsides and other plantings. We're funded by the Iowa DOT through their Living Roadway Trust Fund. So we generally look for things that would be suitable for roadsides. And these species have important functions. Hemiparasites are thought to perhaps play the role of a keystone species in their plant communities. And the 2 species in particular that we are looking at, Pedicularis canadensis on the left is a spring flowering species, and Pedicularis lanceolata, center and right, as a fall flowering species. And their blooming times correspond with the emergence times of the bumblebee giants in the fall, and then their emergence in the spring when they start to establish colonies. Next. So our goal was to not just produce these plants and get them into seed production, but to produce propagation guides and share that information. To start with, we looked for the existing information that we could find, and we went to the Ranger website. If you haven't found that before, Google it. It's really quite useful. There was a plug propagation guide there for Pedicularis greenlandica that gave us some hints. Also, there was a tidbit from Baskin and Baskin about the type of dormancy that Pedicularis lanceolata has. And that suggested it would germinate better with cold moist stratification and that light would improve its germination. But helpfully, and exactly the year that we were just getting started with this, Luke Dahlberg published 2 blog posts on the Grassland Restoration Network. I suggest you take a look at these. They're very good information, and we use these as the basis of our work. Next. So we started greenhouse propagation of swamp lousewort or Pedicularis lanceolata. We had seed that we'd collected from 11 wet prairies around the northern part of Iowa. We stored that seed dry and cold for between 3 and 15 months and then cold moist stratified it for 8 weeks. We were planning on using a longer stratification, but it started to germinate at about 60 days. We had started some host seedlings of 2 sedge species that could co-occur with this Pedicularis species in nature. and then applied 2 seeds of pedicularis per host sedge, or attempted to get exactly that many. You can see the technique we used here of using a plastic pipette to deliver the seeds into a little divot that was right at the base of a host sedge. The seed shown there is actually Pedicularis canadensis, the other species, but this is where I had photos of the technique. Nearly all of the plugs that we seeded in this way ended up with at least one lanceolata seedling in them. One thing that we did find was that some of the vigorous sedges would shade out the seedlings of Pedicularis, and so we ended up having to trim some of the flats in order to get more light to the louseworts. Next, please. We successfully established these in 2 rows out in our production areas. These are plasticulture rows with drip irrigation in them. We had excellent survival of those transplants, and a few of them flowered the first year, and then they flowered abundantly in the second year, which is that picture shown. Next. Once the seeds started to mature, we began to clip the seed stalks as they ripened. We dried them indoors on a tarp, and then moved our little plot combine that you can see in the back of that second image, moved it outside, left it stationary, forked the material into the front of it, and then cleaned it with an air screen cleaner. The first year, there were a few plants that bloomed. We did end up with a small amount of seed. The second year, however, we got a decent harvest. We got 6.5 pounds of seed off of our 2 small plots, which would extrapolate out to about 260 pounds an acre of seed. The second year that second year of harvest, this past fall, that yield was sustained pretty well. Next. Now for the tough one. This is Pedicularis canadensis. This is the one that blooms in spring, and you know spring forbs are trouble. We collected seed of this from 7 prairies in June of 2022. We had stored that dry and cold, which may be part of the issue. We stratified it warm and moist for about 4 to 6 weeks, and then cold and moist for about 10 weeks. This was sort of partly to fit within our production schedule. We seeded 2 or more seeds per host plug. We used both a sedge and 2 warm season grass species for host there, and had very poor germination in the spring that we seeded those. However, we kept those host plug flats and some single species germ flats that we produced with leftover seed. We kept them outside. We overwintered them in an unheated shed, and dun dun dun. In April 2024, we found about 60% of the host plugs had at least one Pedicularis canadensis seedling in them, sometimes more. I think seed moved around during that year outside. And also our germ flats produced a number of seedlings that we transplanted successfully into additional host plugs. Next. And these were healthy plugs. We managed to transplant them out into fields in June, had to do a little watering as a result of that. They flowered in April to May of 2025. We noticed that plants with the sedge host produced more flowering stems, and plants with warm season grass hosts appeared less stressed during hot and dry spells in the summer. Next. We did have these Pedicularis canadensis plants set seed for us in the field. However, the seed is dispersed rather rapidly once those capsules open. The harvest window is pretty short. So my lesson here is check fields frequently. We got a low yield in our first spring and hope to get more seed this year. Next. So our lessons learned are that these are species that can be produced in agronomic production settings. Pedicularis lanceolatus, pretty easy. It has fairly simple dormancy breaking methods. It grows well with sedges and irrigated plots. It's not very susceptible to seed loss, so you can get good harvest, and it's productive at least for 2 years. Pedicularis canadensis is more challenging. It does germinate from stored seed, but that then takes 2 rounds of stratification, and it may benefit from a mixed host stand. Maybe some cool season sedges plus warm season grasses, maybe a legume thrown in there, and has a tricky harvest window. Next. In addition to growing the stuff, we also produce information about it. We currently have a seed production guide in our ebook, Native Seed Production Manual, at our website. For Pedicularis lanceolata, I don't have one produced yet for Pedicularis canadensis. Next. Thank you very much. If you have questions that don't get answered today, my email is there on the slide, and I'm also available at the website, tallgrassprairiecenter.org. So thank you very much.
That was so great. Thank you, Laura. I think we have time for one question. And it looks like the top one is, Laura, what's the theory about hemiparasites as keystone species?
Oh boy, I don't know how great an answer I can give right then, but because they tap into the root systems of their host plants and extract some nutrients from them, it's thought that they may reduce the competitive dominance of some of the other species that they coexist with. And there's some field evidence from kind of observational studies as well as some field manipulations that may indicate that.
Wow, that's awesome. Well,
Yeah.
thank you so much. The pictures were wonderful. No
right. Data graphs though, sorry.
Yeah, that's okay. Well, I think it's time to move on to our next speaker, our last speaker for this session. And it is, let me introduce Jackie Kinch from the San Diego Zoo Wildlife Alliance, talking about seed x-rays. So take it away, Jackie.
Hey everyone, thanks for having me and thanks for the introduction. So today I'm going to be talking about our native plant gene bank and our experience with using an X-ray in our seed processing procedures. Next. So for those of you who don't know, I'm with the San Diego Zoo Wildlife Alliance Native Plant Gene Bank. So we're a group focused on the conservation of rare plants through both monitoring and seed collection and long-term storage in our freezers. We're based entirely in San Diego County, so we just focus on San Diego County species. And as a group, we're always trying to search for new ways to improve our seed processing and our conservation capabilities, including some experimental ones, such as, next, X-rays. So in June of 2024, our bank received a X-ray machine. It's very small scale. It's specifically made to X-ray seeds and small specimens. And we really wanted to try and see if we could use X-rays to help assess seed viability and to determine the quality of our current and past seed collections. And we really liked the appeal of X-rays because they are a non-damaging, non-lethal way to determine viability. So as opposed to cut tests, which would kill the seed, or germination trials, which would lead us with a bunch of seedlings to deal with, X-rays is really quick and easy, and it's entirely safe for the seeds. And so we found that imaging can be used to determine the fill level of seeds, embryo shapes, parasitization, and other anomalies that you can't typically see with the human eye. So as an example, here in the lower right, for those of you who don't know how to read a seed X-ray, bright white means dense, and so in seeds that means filled. So down there, the Cylindra puntia, there's a lot of nice filled, happy seeds. But right in the center there, you can see an empty seed where an embryo never formed, as well as a malformed embryo. Next. So going into some quick learned lessons. So we found that X-rays were really useful for seeds that were larger because they show up better on imaging, and seeds that have hard coats. So in the upper right, that's a Ceanothus species. They form smaller, really hard-coated seeds that are really hard to judge, unless you cut into them, of course. But on X-rays, they show up absolutely beautifully. You can see nice, bright white filled seeds, as well as some empty ones there. We also found that seeds that we would typically sort by color when processing by hand weren't always accurate. So down in the lower left there, we have a picture of an acme swan species and a Malacophalminus species. So if I were sorting those by hand, looking at the real life image, I would typically throw out all of those pale colored seeds, assuming that they were empty, right? But then when we actually X-rayed those accessions, we found that color wasn't necessarily indicative of fill status for our seeds. So with that, it's kind of helped us avoid human error and human prejudgment, where with an X-ray, we can be more sure of what we're doing. And in addition, we also found some fun surprises, which I will share with you now. So next slide. So our first one is parasites. So on the left here, we have a picture of one of our Rus aromatica simplicifolia collections. So they make these flat orange seeds. They have a really hard seed coat. It's hard to judge what's inside. And when we X-rayed them, drum roll please, or next slide. There you go. We found that there were actually parasites in them. So not only can you see where a little larva chewed chunks out of some of the seeds, but you can actually see the little curved shape of a couple larvae sitting in those empty shells there. Next slide. Oh, yeah, there you go. The other thing I wanted to talk about was we actually found twins. So we were doing some germination trials of Torrey pine seeds, and we wanted to do a checkup. And when we x-rayed one of the seeds, we found this kind of strange aberration. So in Torrey pines, that center portion is going to be the embryo, and then the two sections on the side of the cotyledons, and tucked in right next to that embryo is a little tiny sliver of something. And when I saw that, I thought, oh, that might be an actual twin. And sure enough, when we planted that seed in the soil, a few weeks later, we had the main seedling sprout up, and then this little tiny guy sprout up next to it. And this, if you work with Torrey pines, you might know twins do occur in the species. It's not super common, but it is a trend. So it's nice to see that trend reflected in the actual x-rays. Next slide. So getting into some quick challenges we've discovered. So the crucial one is that reading x-ray images can be very subjective. So a good example of this is on the left-hand side, that Tetracoccus species. You can tell there's some seeds that are bright white that we can be pretty certain are good. There's also some that are entirely hollow, but there's all these kind of in-betweens. Some have dark shapes in them, some have weird shapes in them, so it can be hard to tell where to draw that line sometimes. We've also found that each species tends to be pretty different structurally, and so they can have odd quirks, and there can also be abstracting structures to them. So in that middle image, we have Rhamnus species. They have this hard central line in Rhamnus crocea that causes the X-ray to maybe not be as accurate. It causes a lot of shades of gray, and it can be difficult to determine what's filled and what's not. On the right-hand side, kind of an interesting thing we discovered. So that's Chloropyrin species. It's a salt marsh bird's beak. And when we took an X-ray of those seeds, we saw all these white speckles everywhere. So on X-rays, the brighter it is, the denser it is. And so we thought, what could that possibly be? And then when we pulled the seeds back out and took a closer look, those are salt crystals, or at the very least minerals, right? But it's fun to think that we had salt on our salt marsh bird's beak, even if it does kind of ruin the X-ray, unfortunately. Next slide. So overall, just kind of going over how X-rays have helped us out. So we've X-rayed over 60 sessions so far, which is yay, great. One of the main things that's helped us is improving our cleaning methods, like we were hoping for. So in the upper left, we have Monardella viminea. We had a nice cleaned collection of them. We x-rayed them, and then, oh no, almost all of them are empty, right? Only those bright white specks are the filled ones. So after we x-ray, we can kind of look and see, okay, do we need to use a blower or a winnower to try and get out more empty seed? Can we look at color to maybe help us better determine things? We also found that x-rays have helped just overall in our quality assessment. We can be more confident that the species that we are conserving in our bank, we have actual good seed for them for later grow outs and restoration projects. And finally, it's actually been surprisingly helpful in post-germination trial checks. So if we're growing out seeds for a long time in germination trials and they're just kind of sitting there and not doing anything, we can, without disturbing them, take them out, go ahead and do an x-ray and see if they're rotting inside or not. So as a really good example of that, in the lower left, that Torrey pines, we pulled out a seed and x-rayed it, and it turns out it's entirely bloated. And when we cut that individual open, it turns out it was entirely rotted inside. So it's helped us with those germination trial checks as well. Then next slide. I think that's it. Yeah. So thank you to everyone on my plant conservation team. And if there's any questions, I can take them now.
That is so cool to see all these photos. So thank you. Yeah, we definitely have questions. So I'll start with the top one. From Felicia, do you sometimes still get poor germination from seeds that have been scanned and appear to have embryos? I've had issues with Colubrina seeds that have been scanned but still have poor germination.
Yeah, we've definitely had that issue. I think part of it is that sort of human subjectivity when it comes to x-rays that I mentioned, where sometimes we might judge that the seeds look filled on x-ray, but there's a lot of shades of gray. That's where we found the most issue. I think there's probably also an aspect of not knowing how to break dormancy properly, right? So that could be also a conflicting factor.
Totally. And then from Rosa, did you have a process for verifying the X-ray observations, like germ testing, to confirm viability matched with what you thought you were seeing in the images?
Yeah, so kind of similar answer there. We have actually done X-ray, we call them like reliability tests, right? Where we X-rayed a bunch of seeds, split them into good and bad, and then germinated them. And that's been kind of a mixed bag. So a lot of them have proven to be pretty accurate, but there's just some species that when they're X-rayed, whether it's their structure or sometimes if they're smaller or especially if they're flat, they just don't show up super well on X-ray since X-rays are based around density. So it seems like to an extent it's species by species basis for us.
Makes sense. And I think we have time for one more from Scott. Are the X-rays digital and what is the cost per image?
Yes, the X-rays are entirely digital. The cost, it's really just the machine. So machine it looks like a computer tower. It's pretty small. It just sits on top of a desktop. Unfortunately, the machine itself is very expensive. We got it through a lump donor to the organization, which was very nice of them. And then there is quite a high maintenance fee for the machine itself. So yeah, it can be a little cost prohibitive, but it's not like on a per image basis for us.
Well, this was a great talk. So I think we're done for now and we'll go ahead and take our break. But thank you so much, Jackie.
Yeah, of course.
Really appreciate it. And, you all can find her if your question did not get answered. Go ahead and find her in Whova and ask your questions in that agenda session as well. So thank you so much. For everyone still on the call, let me stop sharing real quick. This is the main Zoom session. So when you come back for the next